In this talk, we are going to discuss the basic steps in the construction of phylogenetic trees, and one of such methods, the maximum parsimony method. The process of carrying on sequence-based phylogenetic analysis can be divided in 4 key steps. The first step is the selection of a sequence of interest that we want to investigate. This could correspond to a protein sequence, or a gene, or a whole genome. The second step is to identify homologs. Thus, we need to acquire sequence data from sequences that are homologous to the sequence of interest. The third step is to align the sequence of interest and the homologous. Finally, we calculate the phylogenetic inference from the alignment of the sequences. Which kind of sequences we could use for build a phylogenetic tree? We can use a complete gene sequence, or sequence from coding regions, introns, promoters or intergenic spacers, whole genomes, proteins, or functional non-coding RNAs. Once we have selected our sequence for study, homologous sequences could be obtained from different methods. One is obtain the sequences of interest from biological samples, which could be amplified and sequenced using specific primers in the case we are interested in a particular gene, or using a random method, using a shotgun sequence approach if we are interested, for instance, in comparing whole genomes. Another way to obtain homologous sequences is by searching in sequence databases like GenBank or UniProt from protein sequences. In this type of searches, a decision must be made among the resulting matches as to which are truly homologous and which are not. This step is critical and the source of many problems. Similarity of sequences in some cases is not proof of homology. Sequence similarity could be a reflection of homology, but this also could be a result of a convergent or parallel evolution. So, we have to examine carefully the statistical parameters of the resulting search, like the bit scores and e-values, to be sure that we are selected for comparison to homologous. The third step, and perhaps the most important, is the alignment of the selected sequences. In this sense, we have to be careful about some issues. For example, when distantly related protein coding genes are being compared, alignments can become more challenging. A key problem is the occurrence of insertion and deletions mentioned above. The more insertions and deletions that have occurred, the more difficult it will be to identify homologous position within genes, and this could be a problem. In other cases, aligning homologous non-coding region is quite challenging, because the patterns of conservations are very different from those seen for a gene. This means that algorithms for alignment that has been designed to work well for gene sequences may not work well for many non-coding regions. Thus, we have to be sure of the quality of our alignment, because if it's not accurate, the resulting phylogenetic would be incorrect. Once a multiple sequence alignment is in hand, a phylogenetic reconstruction method or methods must be chosen. The many and remarkable diverse methods can be classified into 4 main categories on the basis of their overall schema. These methods are parsimony methods, distance methods, maximum likelihood methods, or Bayesian inference methods. Let's take a look to parsimony methods. Parsimony methods are based on the principle of Occam razor that states that, other things being equal, the simple explanation, or the most parsimonious one, should be the correct. In parsimony methods, you compare all possible trees that generate a set of alignment sequences, and each is given a score that is equal to the number of character changes that would be required to produce that tree. The optimal tree is considered to be that which requires fewer changes, the most parsimonious one. As an example, imagine that we have a DNA sequence from 5 taxa. In a parsimony analysis, our goal would be to generate all the possible trees showing the relationships among the 5 taxa, and then to determine the number of evolutionary changes that would be required to generate the sequence alignment given each of those trees. To make this very simple, just consider one alignment column, this column, and 2 possible trees. For each of these trees, we need to determine how many evolutionary changes would have been required to produce the observed character states in column 1. So, we have our trees representing the different combination of character states. And we reconstruct the ancestral characters that could give rise to the observed character states. These are placed in the ancestral nodes. We have 2 G's. The ancestor is a G. Here, also the ancestral character is a G. Here, we have an A, so the ancestor has to be an A character, and similar from here. In this case, supposing that this is the ancestral character, we have to be a change here and a change here to produce an 2A characters here. Similar for this other tree. If we want to reproduce this, we have to change here and also here, and the same for these three. When a change is required to modify that character of the ancestral node, we indicate with an arrow. Here we need to modify here and here, 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 and here, and here. Finally, we count the character state changes in all trees and select the tree who require less changes. In this case, this tree, only with one change, you could obtain the observed character states in the first column of the alignment. We do this for all columns of the alignment and score all trees according to the number of character changes they require, and select the one who requires less changes, the most parsimonious tree. This is basically how it works, the parsimony methods. Parsimony methods is a very simple approach and is popular for that reason. One major drawback of the method is that as the number of taxa goes up, the number of possible trees increase exponentially. Thus, could be very time-consuming when you process large datasets. Another problem of the parsimony method is known as long branch attraction. This phenomenon occurs for certain species that evolve under convergent evolution, which means that a specific character could evolve to a similar structure in this species, but they not share a common ancestor. This process is also called homoplasy. And as you can see, these two taxa, they share a similar character, but this character doesn't come from the common ancestor. In this sense, the parsimony methods could incorrectly group together these two species in closest branches, and this could give an incorrect alignment.